Welcome to the Faculty Focus podcast, live from our annual Faculty Research Day with your hosts
Marleen
and
Teun,
highlighting the research of our professors and PhD researchers.
Next on our podcast, we have Olivier Beyens. His topic of today is computational design of DPP8 and DPP9 inhibitors using cosolvent molecular dynamic simulations. Welcome to the podcast, Olivier. Can you briefly introduce yourself to our listeners?
Thank you, and yes, sure. So I'm Olivier. I started my academic trajectory in 2015 with a Bachelor of Science in Engineering Science, and then 2018 I chose a specialization in chemical engineering. So then I finished my master's degree in 2020, and that's when I started my PhD degree here at the University of Antwerp at the Department of Pharmaceutical Sciences at the Research Group of Medicinal Chemistry in the topic of computational drug design.
Quite some trajectory already.
Yes, thank you.
Yeah. Well, imagine that we have a teenager here in the room who's fascinated by science and wants to know what you do all day. How would you explain your research?
Well, how I would explain it is that I use computer simulations to design the molecules that could be used as drugs in therapies. That's how I would explain it in simple terms.
In simple terms, but clear terms indeed. You work with cosolvent molecular dynamic simulations in your research. What would you want us to understand from that concept?
So how it works is that, you would take your target protein structure and you would place it in a virtual box. Mm-hmm. And that virtual box you will fill with waters and then some small chemical fragments. And those small chemical fragments are then called the cosolvents.
Okay.
And why you want to do that is because if you choose those cosolvent molecules in such a way that they represent common drug binding features,
mm-hmm.
then you can get a kind of map of where they like to reside in the protein structure during the simulation. And that way you can see, okay, this chemical binding feature is important in that location in the protein, and this chemical binding feature is important there. That's basically the goal of the simulation type.
So you can see where they connect with the protein.
Yes, in a way, yes. They will not connect covalently, but they will form what we then call intermolecular interactions.
See where they like to sit on the sofa.
Yeah, indeed.
Something like this, all right. And the actual cosolvent part is a part that you add the additional chemical structures?
Yeah, so the additional chemical fragments, we call them. So they're very small molecules mm-hmm. with, for example, an example would be, for example, isopropanol, so that you have mm-hmm. one hydrogen bond donor and acceptor group. That could be a certain part of cosolvent that is commonly used.
Okay, and that's different with what's been done before, because before they used water and protein?
Yeah, if you do water and the protein, that's a regular, what we call
mm-hmm.
then a regular molecular dynamic simulation. So the cosolvent is really that you add those isopropanol molecules. And it's a relatively common technique in computational drug design, but we then apply that to our targets, which are called then DPP8 and DPP9.
All right, okay. And would you say that, in wet labs, a lot of things can go wrong? What do we have to think about when things can go wrong in a simulation? Or?
Well, things can definitely also go wrong in a simulation. I can assure you that.
Yeah.
But the nice thing about it, I think, and the difference with wet lab work is that it's easier to figure out what is going wrong. I think, because mm-hmm. it's usually related to some kind of obscure setting, or you can always go. Yeah,
something like that.
Yeah, in a lab you could sneeze, but on the computer that's also not a possible thing that can go wrong, of course. But if a computer simulation is going wrong, then you can still a lot of the source code is also available online, and you know a bit the steps that are going on. It's a bit easier to conceptualize, I think. Mm-hmm. So it's a bit easier to figure out where it's going wrong and rationalize why it's going wrong. I think that is what makes it a bit easier to troubleshoot, let's say.
All right. Than in the simulations itself. Okay, you work with very specific targets in your research. Is the list of targets you can use endless, or are there factors that are very important when you have to choose a target to work with?
When you choose a target to work with, what we usually do is we hope that there is a crystal structure available of that target with a high resolution, because then the crystal structure is a bit more accurate.
In real life, you mean, or that you can put in the simulation then?
How that works is those crystal structures are determined by experimentalists, so mm-hmm. it's lab work. Okay. And then usually they are they are put on a data bank, and we can use that data bank to look for structures to put in our simulations.
Okay.
So what we look for is then what we preferably available is a target with a good high-resolution structure.
Okay.
Nowadays, you also have the alternatives like AlphaFold-generated structures.
From the Nobel Prize.
Yes,
Yeah.
exactly. But yeah, there the problem is, you're also always a bit less certain about it because it was produced by a model. So then you may want to do some additional pre-processing steps before starting your simulation.
I know that one of your supervisors works with a VR in a class he teaches. Are there any other VR possibilities or AI possibilities in your research?
Yes, so yeah, it's an upcoming field. So also in computational drug design, of course.
Mm-hmm.
So what now some research attention is going to is to also use those AI models in drug design. So they can have many different uses. They can be to try to predict affinity or to try to generate new molecules that can serve as drugs. So then you're looking at generative AI models. But yeah, the problem with those is that we're still not there yet that it can be used at a really high accuracy. For example, if you compare them to, let's say, what we then call the physics-based techniques revolving around molecular simulation, they often have a higher accuracy, but also at the cost of a bigger computational expense usually.
Yeah, so you're not saving time yet with AI tools.
You can. The thing is that you can try to save time with AI tools, but you might lose some of the accuracy.
Okay, all right. Well, maybe something a little bit more off topic, not really, but more general. Why is it important to share research results? Because you mentioned, for instance, that you made computed qualitative fragment affinity maps freely available. What could this mean for other researchers?
Yeah, so what those maps are, that's basically the output of those simulations. And so that was where the molecules like to reside on the sofa. So those maps of where they like to sit on the sofa.
Cushions,
that we released publicly. So for example, if you were also interested in DPP8 or DPP9 drug design, you could look at that map and try to see, okay, I now have this inhibitor structure. How could I optimize it further? And you could try to gain some inspiration from those maps. For example, here, okay, there was that isopropanol molecule. Here we need to place a hydrogen bond donor or acceptor.
Did you also use other researchers' maps for your own research?
No, because as far as I know, we were the first ones to compute these cosolvent molecular dynamics maps on DPP8 and DPP9.
All right, I have to say, hoo-hoo for this, right? Very well done. A cheer, yeah, true. Is there something else about your research that you'd like to share that we didn't ask?
Yeah, we also did perhaps some tool development with regards to the setup, mostly of related to the setup of some specific type of cosolvent molecular dynamics simulation. And that tool we also made freely available. So I think that's a nice example of that the output of academic research is available to the wider public for free. And what else we did? We also there was also another technique that you can use to computationally optimize inhibitor structures, and that we also used on the DPP8 and DPP9 targets.
All right, very interesting indeed. Is there some advice you would like to give to new starting PhD researchers?
That's a good question. I would say that, well, for me personally, the first year was perhaps not the most fun year because I was placed in also a bit of a new field, and you have to learn a bit also
problem solving.
Yeah, the simulations are crashing. You don't know why yet, but at a certain point, I think somewhere after the first year, you gain more experience and you start to understand. Okay, now this is going wrong or that is going wrong, and you can more easily solve it. So
you persevered.
Yeah, and the tip I would give is to also maybe take time. The PhD is 4 years, so you have, in a sense, a lot of time to sometimes take some time and investigate deeper into a certain subject that is of interest of you. That I think that you gain some more background knowledge in certain areas. I think that can be a useful tip.
And also helpful for later in the process, of course.
Yes, indeed. Yeah.
All right.
When will your PhD be finished?
Hopefully next week, Tuesday. Then it's my public defense.
Next week's Tuesday?
Yeah.
Okay. So it's already finished. You're just preparing for the public defense then?
Yes. So I made my presentation yesterday and today, so hopefully I will successfully present it on Tuesday.
Well, if you answer as well as here, then it will go whoops.
I hope so.
Sure.
Many thanks for coming by.
Yeah, thank you.
It was my pleasure.
Yeah. Do you have already an idea where you're going?
Yes, I will be the 1st of November. I will be joining the VIB Discovery Sciences unit. So they try to do computational. Yeah, they try to do drug discovery for spin-offs of the research group related to the VIB.
Congratulations.
Yeah, thank you.
Sounds very interesting as well.
Yeah, I'm looking forward to it.
Okay, good luck anyways.
Thank you.
Thank you.
Bye.
Bye.